I'll do a quick intro for Amy and Donna. They do work out of the Ohio Water Science Center, Ohio, Indiana, Kentucky Water Science Center for USGS. They've been doing this work on using the virtual beach modeling software in conjunction with monitoring data, and then using the model to predict the probability of either harmful algal bloom in at least one case and E. coli above target levels in many other cases. And I'll let them take it from there.
Can you hear us now?
Yes.
Excellent. Thank you, Alex, for the introduction.
You're not getting any feedback or anything?
We hear you loud and clear.
Okay, great. Okay.
All right. So, again, thank you for letting us talk to you today. I'm Amy Brady, and my co-presenter is Donna Francie, and we're both out of the Columbus, Ohio office of the USGS's Ohio, Kentucky, Indiana Water Science Center. So obviously we're going to be talking to you a little bit about our Nowcast system to help with management decisions, specifically for waters are affected by fecal contamination or cyanobacterial harmful algal blooms. And we'll be referring to that as cyanoHABs throughout the talk. Just another. There we go. That does it. Okay, good. All right, so our agenda for the talk will be to provide some background information on modeling for fecal indicator bacteria and for cyanoHABs. We want to discuss some suggested types of data to collect for the cyanoHAB models, showcase some examples of some site-specific models, and then discuss our Great Lakes Nowcast system. And I also want to point out our disclaimer that I have to put here, just to mention that our data shown in this presentation are still preliminary and subject to review. So as you guys are probably all aware of, this problem that we're facing with bacteria and measurement of toxins is this delayed notification. For culture methods, the results are only can be available in 18 to 24 hours. If we're measuring toxins, which can occur in some of the cyanoHABs, these results could take several hours before we're able to get results. So this delay obviously means that we're putting people at risk of coming into contact with these pathogens or toxins. And so we need to find a way to provide more timely results to decrease this risk. So a Nowcast can be used for this purpose. So a Nowcast is a system that we use to inform the public of estimated water quality conditions in near real time. We use simple mathematical models based on easily measured environmental or water quality variables to make these management decisions. For example, a beach Nowcast can be used to inform the public that based on conditions at the beach today, the beach manager has decided the water quality to be good and open to the public. Alternatively, if the conditions indicate that the water quality is poor, the beach manager can issue an advisory or closure. So how do we develop our Nowcast? We specifically use multiple linear regression models to develop site-specific models. So multiple linear regression models can contain two or more, as I mentioned, these easily measured or observed variables that are related to our target concentrations. As an example, this is the stream flow on the Cuyahoga River, which is on the x-axis, related as compared to the E. coli concentrations, which are shown on the y-axis. So you can see that there is this relationship between these two variables. So we want to collect data over multiple years to produce a data set that has these varying hydrologic and water quality conditions. As you can see in this graph, the relationship between stream flow and E. coli has changed over time. And so if we're able to incorporate these different water quality and hydrologic conditions in the model, we can produce a more robust model. Once we develop the models, the output is either a predicted E. coli concentration or the probability of the concentration exceeding a specific threshold, for example, a water quality standard. We use US EPA's virtual beach software to create our multiple linear regression models. This is free software. It's available at that website that is shown here, or you could just do a Google search for US EPA and virtual beach. It was originally designed to predict E. coli concentrations at beaches, but we can use it for any dependent variable or water body. For instance, we can look for a toxin in a river as example. Virtual Beach is a great tool. It allows for data exploration, model selection, development, and validation. In addition to the multiple linear regression models, users can also choose partial least squares and generalized boosted regression modeling. These are a couple more options that are available to fit their data. But all the models that we're presenting in this talk were created using Virtual Beach. We started working on Nowcast over 20 years ago, and we have a pretty solid understanding of what we're doing, I guess. But we've steadily improved on our methods and our capabilities. The Ohio Nowcast started operationally in 2006 with Huntington Reservation Beach in Bay Village, Ohio. This was our first site. In 2011, we started to use Virtual Beach specifically for all our modeling, which was a great improvement over our previous modeling methods. And then the Great Lakes Nowcast, which I'll discuss in more detail later in the presentation, started in 2016 with the joining of the New York and the Ohio Nowcasts. And in recent years, we were able to expand the Nowcast into Pennsylvania and to Michigan and start research for modeling cyanoHABs, thanks to the Great Lakes Restoration Initiative funding. We've produced several reports, and our team, not just us, I should say, have published several reports. And if you're interested in any of these, you can contact RI for that information. For E. coli models, we develop what we're calling real-time models. These real-time models include variables that are easily or continuously measured. We can quickly access this data. So the variables that we found most useful for these models include turbidity, wave height, 24-hour change in lake level, numerous weather variables including rainfall, wind speed and direction, and the barometric pressure, and then daily mean stream flow from local stream gauges that are affecting that specific site. The graph shown here is, again, the Cuyahoga River data. It's E. coli on the y-axis and turbidity on the x-axis. And this data actually is for 10 years of data collection. And obviously, turbidity has a very strong relationship to E. coli, and it is to be the main variable in the Cuyahoga River model. So, how do we judge whether our models are doing well or not? Obviously, this is important. We don't want to, make an improvement if it's not really an improvement. So to understand, how well we're doing, we're going to compare our Nowcast models to the traditional method of using the previous day's E. coli concentration. So basically, if we collected a sample yesterday, we get our culture method results today, and that is the data that we would use to post an advisory or a good day on that at the location. This is the traditional method. So this table displays the results of the Huntington Reservation Beach in Bay Village, Ohio, which is a Lake Erie beach near Cleveland. As I said, this is our first beach, so we do have a lot of data from Huntington. So our model was able to correctly predict whether the water quality standard was exceeded, not exceeded, in 84% of the samples that we've collected, whereas the traditional method did the same for 74.5%. A couple other statistics that we look at, though, just to make sure our model is doing well, is the specificity, which is how well our model is able to predict a non-exceedance or a good day. So the Nowcast model, obviously, it did a little bit better than the previous day's E. coli. The last statistic, which I think is the most important, is the sensitivity. And this is how well our models correctly predict exceedances of our water quality standard. So we want to be able to predict these exceedances because we want to be able to tell the public whether if they would be at risk if they were in the water and possibly in contact with pathogenic organisms. So as you can see here, our Nowcast model had a much higher sensitivity than our traditional method. And so that implies that by using the Nowcast, we are decreasing the risk to the public who use this beach. Now I'm going to turn it over to Donna so she can talk more about the cyanoHAB modeling.
Okay, thanks, Amy. So cyanoHABs, they've been for several years a major water quality issue for Lake Erie, especially the Western Basin, and also for our inland lakes in Ohio. You all probably heard about the infamous City of Toledo had to issue a do not drink advisory in 2014. So around Ohio, we've been working really hard to address this issue. And water managers have proactively turned to the HAB bulletin, which is on the left here. That's a bi-weekly forecast based on remote sensing. It's put out by NOAA, and it helps. It helps the drinking water treatment plants. It helps the beach managers somewhat to give them an idea if a cyanoHAB or a bloom is heading toward their direction. However, microcystins, which are the main toxin of concern out our way, they're not pigments, and they cannot be directly detected by remote sensing. So another solution that we've come up with to augment the remote sensing is to estimate the toxins themselves in regression models. So Amy talked about using real-time models for E. coli. For cyanoHAB toxins, we've come up with two different types of models. The real-time models, which include factors that are easily or continuously measured. And then the second type of model is a comprehensive model. And that uses factors from samples collected and analyzed in a laboratory. Now, these take more time and effort because you have to send the sample to the lab. However, they provide more information about maybe the factors affecting the community structure, what affects toxin production, and hopefully can provide an advance warning of a toxin event. And here are some of the examples of the real-time and the comprehensive variables that we've been working with. So we've been working to develop cyanoHAB models at drinking water intake and recreational sites. And this is our latest project, started in 2016. And the goals in this project were to first identify factors related to microcystin concentrations. And then if we're able to identify those factors, develop and implement the use of multiple linear regression models to estimate those concentrations. Here's a map of our sampling and data sites. The recreational sites are the pink dots, and the green dots are the drinking water treatment plant sites. There's 4 drinking water treatment plant sites and 2 recreational sites associated with that number 1. That's in the western basin of Lake Erie. And then with the number 2, those are our inland lake sites. Those are in northeast Ohio, and those are both drinking water treatment plant sites. So we collect a lot of ancillary data for use in our models, and those are the other symbols here. So the stars indicate locations of continuous water quality monitors. The black triangles are USGS stream gauges. The blue triangles are lake level stations, and the yellow crosses are National Weather Service airport weather stations. So we use all these data sources for our models, and they're all readily available. So whether we collect our samples from a tap in a drinking water treatment plant, or a wet well, or a composite sample from a recreational beach site, collect samples during the cyanoHAB season, which for us is generally, we start in May, and things really start to increase kind of July and August. And they can actually, in our inland lake sites, go on till about this time of year, to early to mid-December. Samples are analyzed for microcystins, for nutrients, for cyanobacterial genes. Those are our comprehensive variables. Physical parameters, either handheld, sondes, or continuous monitors, and then the environmental data I showed you in the previous slide. So we use several assays for cyanobacterial genes that we analyze by quantitative polymerase chain reaction, or qPCR. And the USGS assays target general cyanobacteria and then the 3 genera of cyanobacteria most commonly associated with microcystin production in freshwaters: Microcystis, Silicospermum, and Planktothrix. So we also target the gene required for microcystin production, the MCYE gene, and identify which genus is associated with that gene. Now, Ohio EPA, who has a large program, requires the drinking water treatment plants to collect samples every other week during the season for general cyanobacteria, a general MCYE gene, and saxitoxin gene. Really important to developing models for cyanoHABs is a network of sondes that we have in the Western Lake Erie Basin, and it's coordinated by the Great Lakes Observing System at drinking water treatment plant intakes and also other locations. Plus, there's some private service sondes at our inland lake sites. So for the continuous monitors, data are collected in 10 to 30-minute intervals, depending on the site. And we don't want to rely on one measurement of pH, specific conductance, phycocyanin, or whatever we're measuring. Instead, what we'll do is we'll take an average measurement for the 24-hour period up to 10 a.m., which is the approximate time the microcystin sample is collected. Then averages are calculated from longer periods, so antecedent to the time the sample is collected for 3, 5, 7, or 14 days. So it requires a substantial amount of work to compile these data, and we have a spreadsheet that Amy designed for doing this that really, really helps. If anybody is interested, contact us. Third-party environmental data, these are all available on the web, and we compile them for various time intervals. Now, for E. coli that Amy talked about, we only have to compile up to 3 days antecedent to the time the sample is collected. However, for the cyanoHAB models, we've noticed longer time frames are needed, up to 14 days for rainfall and lake level, and 30 days for discharge. So you can see the different types of data manipulations that are done. Okay, so now for some case studies, and I'm going to present data from 2 sites today to give you an idea of where we're heading with this research. So the first site is Maumee Bay State Park. It's a popular recreational area with 2 beaches. It's in the western Lake Erie Basin, and it's just east of Toledo, Ohio, and it's also 5 miles east of the mouth of the Maumee River. And the Maumee River it drains a large watershed. It's a huge river, 5,000 square mile watershed, mostly agricultural land use, so that's where our nutrients come in and why we have a cyanoHAB problem in the Western Basin. So we've been working with the University of Toledo at the park since 2004, and we're still working there with them. I'm going to present the cyanoHAB data for 4 years collected at the Lake Erie Beach. So the first step in developing models is to identify factors related to the microcystin concentration. And so we generally do this using Spearman's correlations or Spearman's rho, which measures the monotonic relation between two variables. So y generally increases or decreases as x increases. So in this case, and what I'm showing you is the relation between microcystin and other variables. And those relations that were significant at p less than 0.05 are indicated in blue bolded text. So the first 4 lines there are data on water quality parameters. There's 2 columns. There's a handheld measurement done at the beach at the time of sampling, and also the continuous monitor measurements. And these are from 3 different nearby ponds with 5 different time frames, 1, 3, 5, 7, and 14 days. So it's a compilation of data. And you can see that the phycocyanin and the pH were significantly correlated with microcystin, with the handheld measurements having a higher correlation than the continuous measurements. Let's see. And then we can move down. Specific conductance was weakly negatively correlated, and water temperature was weakly positively correlated. The Maumee River stream flow is negatively correlated to microcystin concentrations. And also not shown here were some variables that were not significant: lake level change, rainfall, wind speed, solar radiation. Chlorophyll was not significant all the time, and wave height. NOAA satellite data had a weak correlation to microcystin. We requested these data from NOAA, and Steiner coastline of day of the year, which is, a seasonal variable, was also correlated. I do want to say that correlations do not provide evidence for cause and effect. One may cause the other, or they may share the same cause, but evidence for causation must come from outside the statistical analysis and knowledge of the processes. So you can't get everything you need from correlations, and the next step is to make lots of plots with the independent variable on the x-axis. In this case, it's phycocyanin, which is a pigment produced by cyanobacteria, and microcystin is on the y-axis. The handheld measurements are on the left, and the 14-day continuous average is on the right. So both are significantly correlated in microcystin, although there's a higher row for the handheld measurements. And we want to look at these plots and make sure that the relations are not influenced by 1 or 2 data points, identify any outliers, and ensure that the relation holds up from year to year. Okay, so I'm going to show some preliminary models. These haven't been finalized or approved, but I'll give you an idea of what may work for modeling, estimating microcystin concentration. So this is a real-time model using data collected at the site, and in particular the phycocyanin handheld measurement, and also some third-party data, the discharge, and then data that, of course, are always available, which is cosine of day of the year. So this model had 78 data points in an R-square of 0.6, which is not too bad, not great, but not too bad. So if one were to use a proposed recreational standard for at an advisory of 4 micrograms per liter, and that's this green vertical line. That's actually the log of 4, is that green vertical line. And you could set a threshold for this model of 48% probability, which is the horizontal line. So in other words, you would have a 48 chance of exceeding that threshold of 4 micrograms per liter. So Virtual Beach will graph this for you, which is really handy. And it has the observed microcystin concentration on the x-axis and then the probability on the y-axis. And what it'll do, it'll put the data into 4 quadrants, and you want to maximize the data in the upper right and the lower left quadrants, which are your correct non-exceedances and your correct exceedances. In doing that, we can raise or lower our threshold, and we set it at 48% to get an accuracy of 82% for this model. Okay, so that's Maumee Bay State Park. Our second site is the Caddis Water Treatment Plant. So the village of Caddis in right here in northeast Ohio pulls water from Tappan Lake. It's an inland reservoir regulated by the U.S. Army Corps of Engineers. It's a popular recreational lake. And I'm going to take a look at comprehensive variables at Caddis from 2016 to 18. So it takes time to get these laboratory measurements of nutrients and genes. So what we do with the comprehensive variables is we want to use them as an early warning. And to do this, we have to take the last gene or the last nutrient concentration 1 to 2 weeks ago and determine the correlation with the current microcystin concentration. So we lag the data up to 2 weeks. And if we go ahead and do this, among the nutrient constituents, nitrate plus nitrate was weakly negatively significantly correlated with the microcystin. None of the other nutrients were significantly measured: orthophosphate, total nitrogen, total phosphorus, and ammonia. But however, several assays were used to analyze samples for cyanobacterial genes by qPCR, and the Dilocospermum general gene had a pretty strong negative correlation. The Planktothrix MCYE gene had a positive correlation. Planktothrix is one of our major toxin producers in this inland lake. And also the microcystin general MCYE gene had a strong positive correlation. So we can go ahead and take a look at an example of a comprehensive model for the Caddis Water Treatment Plant. And this model has the microcystin MCYE gene lagged, of course. Lake levels change from 7 days ago, a day of the year variable, and also a rainfall 14 days sum variable at a nearby airport rainfall station. So we had 97 data points, an R-square of 0.61, accuracy of 87%. So it does pretty well at this inland lake site. Okay, so what we have to do next is take these models and validate them during an independent year and adopt them into the Nowcast that Amy is now going to talk about.
Thanks. So in the Great Lakes region, the Great Lakes Nowcast is used to provide some near real-time beach closures and advisories. We have federal, state, and local agencies working together to make this Nowcast system work. And each of the operational Nowcasts in this system use multiple linear regression models to calculate either their predicted E. coli concentration or a probability that that water quality standard will be exceeded. So there are 2 portals as part of this Nowcast system, the public portal and the database portal. Yes, I want to say portable for some reason. Both of them are accessible using smart devices, so I guess they are portable as well. Okay. So the public portal provides the public with some valuable information about the Nowcast, including the daily water quality predictions. And the database portal is only accessible to site managers and was developed to increase speed and efficiency of modeling. So this Nowcast, the system is very versatile, and it can be expanded to include new sites and adapted to include additional water quality threats, such as the cyanoHAB. So this is the main page of the portal, or the public portal, and it is a mapper that shows the Nowcast sites across the Great Lakes. Actually, we're kind of hiding some behind that legend there, but that's okay. There's some more up there in Lake Ontario. So the Nowcast sites, they're shown as these colored balloons, and they indicate the current water quality conditions. So green means good, and then the red or black balloons mean an advisory or a closure is present. But if you click on an individual balloon, some site-specific material is available. There's several different little tabs under this window that pop up. So for the current conditions tab, you can see the predicted E. coli, the predicted concentration, or excuse me, the predicted conditions. This was pulled, during the off season, so it kind of shows you that it says off season right now. But if it was during the recreational season, this would either tell you it was a good day, an advisory day, or a closure day. It also tells you the current water quality temperature, or excuse me, the water temperature, and provides you this map of the site and the possibility of getting those driving directions if you're looking, if you're in the area and looking for a beach to go visit. If you click on the site details tab, you'll get some more information regarding the specific site, who's managing the site, specific contact information, and the models used in the variables used in the model. If you click on the recent conditions tab, the public can quickly access how well the predictive model is doing and compare it to the traditional method of using the previous day's sample concentration to estimate current conditions. So the database portal, as I mentioned, is only available to site managers and it requires a login. However, field technicians can enter field parameters directly into the database while in the field. We also enter our lab results and our model results so that this data can be sent directly to the public portal for display. One of the most useful features of the database is that the system harvests data from online sources, such as the National Weather Service, NOAA, and the USGS, and stores this data to allow managers to use these data in their models without having to search out this information manually. This is a big improvement over what we've done in the past, where we've actually had to go out ourselves and to the different sites and harvest our information. Kind of as a summary, there are many benefits of nowcasting. Our nowcast can provide near real-time swimming advisories to the public. Water treatment entities can use nowcasts to trigger sample collection and use the data to optimize water treatment and intake options for the current conditions. The cyanoHAB comprehensive models can provide advanced warning of a potential toxin cyanoHAB, as well as provide additional information to help understand what factors are related to toxin production. Without our partners, none of this work could have been done. Federal, state, and local governments, drinking water treatment plants, and university partners that help with sample collection, project planning, and data management are crucial. And again, the support for the project, I've mentioned the Great Lakes Restoration Initiative, but also the Ohio Water Development Authority and the Muskingum Watershed Conservancy District. So, thank you for your time, and we would welcome any questions that you may have.
Thank you very much. Open up to the floor to questions. If you're online and would like to ask a question, just unmute yourself and ask your question. We'll start in the room. Does anybody have any questions, comments?
So I have one sort of question. Could you guys comment a little on like the minimum data requirements to build this type of model? I know you guys have a wealth of information from a lot of those beaches, which we don't necessarily have, but
yeah, we typically recommend for at least for the E. coli models, at least 2 years of data to help build the model, and use that third year to kind of validate to make sure it's working well. The cyanoHAB data, a little bit more Go ahead, Donna.
2 years of data. So the E. coli, you sample what, about 3 times a week? The cyanoHABs, if you have weekly data between May and December and enough, you know, high microcystin concentrations, if you have 2 years of data, you could develop a model after that.
Do you guys know how many people are making management or like beach advisory decisions based off of these models?
Well, at these sites, people are making management decisions.
Okay.
People are logging on to the portal, the public portal. Do we have counts on that?
Not off the top of my head,
Yeah.
I can get some.
Yeah.
Yeah, so I mean, and it's not just the Great Lakes Nowcast either. There are several other types of Nowcasts work across the United States.
In California, in Rivers, in Philadelphia, in Georgia, they do it. South Carolina.
Mm-hmm.
So they're actually like going out posting signs based off of what the model says?
Yeah, or updating a website. Yeah. At some of our sites, we do post signs. At others, do they post signs everywhere?
I think at the majority of the beaches they do. The Cuyahoga River, they just have a website link that you can link to the Nowcast site and get that information.
Okay, thank you.
We're actually putting a review paper together, and we're trying to find out where models are used for management decisions around the country. And we're getting pretty good response. They're used.
Okay, thank you. Yeah, I think I got that email, so.
Oh, you got the email. Okay.
I'm looking out for that.
Okay, thanks.
We have time for additional questions, comments? Folks online? And then Ken, you sent in a question via the chat box. Yes, we will have the presentations available on the work group's website. We are recording, so I will try to edit the presentations and get those up as well. We'll have links on the website, and they'll be hosted on the YouTube channel for the collaboration network. It's another working group for the council. I think you guys nailed your presentation. We don't have many questions coming up.
Is there someone?
Yeah, I was gonna say, is the model like the model structure behind it, like how to create our own type of predictive model available or published publicly? So if California was looking to try to adopt a similar model, is that doable if we collect the data?
You mean instructions on how to model?
Or no, just the model, you know, the structure. So like if we collected the 3 years of data and we're looking to create a similar or the same model, to be honest, but with our own data, is that publicly accessible, like on a GitHub or something like that? Like is that something that's doable?
Well, yeah, you wouldn't want to use our models because we're in Ohio and you have different conditions out where you are. However, we are publishing a paper, and all of our models, we have 18 models, they'll be available through Sciencebase, which is a public database, you know, that we're required to do, that federal government employees are required to do. So all the models will be there, the virtual beach files, everything, when it gets published.
Great, thank you.
Yeah, this is Heal the Bay has a GitHub where we kind of show you how to do a model as well. So I'll somehow get you my contact information. Yeah, we should have a chat about that.
We also have done some training for using Virtual Beach and modeling that way, and so we do have some training materials. Those aren't published anywhere, but if you are interested in them, you can contact us and we can send that out to you.
Yeah, the basic bare bones for Virtual Beach, you know, Nowcast is available at USGS and EPA, right?
Yeah.
Yeah.
The software itself is available at US EPA. It's free. Is that what you mean?
Yep.
Okay, yes, yes. And we do have, like Amy said, these training modules. We're not doing a training class this year, but we do have the modules, and we could definitely share them with anybody that's interested.